Okay, last lecture. We're going to talk a bit about chromosome organization and just a little bit about the phenomenon of imprinting, just so that you know what it is and something about it, not in any great detail. Okay, so the levels of chromosome organization, some of which are familiar already and others not. We, of course, have the DNA, which is complex with the histone proteins to form nucleosomes. Okay, next, and we'll talk about these somewhat in order, we have the topologically associated domains, in which chromatin is organized into loops, which will essentially keep them sequestered from one another in terms of their interactions. Then we have compartments A and B, which are active versus repressed, and then we have chromosome territories, where each chromosome actually occupies a discrete region of the nucleus. So there's a very high level of organization here, a lot of which is not really understood very well at all. So here's a picture which summarizes it. We have a picture of the nucleus, and the little colors, the different colors, indicate the locations of different chromosomes. So this was done by a form of immunofluorescence with probes specific for each of the chromosomes that were distinguished according to the colors that could be interpreted. So we can see that each chromosome has a little separate color and occupies a separate location in the nucleus. It's not all mixed up like spaghetti. Okay, then we have the A and B compartments. And again, there's not much understood about these quite yet, where the A compartment is where the active chromatin is, and the B is the repressed. Okay, next we have the TADs, and we're going to talk more about those in a second. Those are the topologically associating domains, in which the chromatin is organized into loops in order to isolate one from the other. Okay. Finally, we have individual chromatin loops, and at the lowest level, we have, of course, the nucleosomes. Okay, here's another picture of a chromosome territories as determined by Fish. The chromosomes are painted different colors using different combinations of fluorescent compounds, as before. So we need to mention the A and B compartments. The A compartment includes active genes. The B compartment is the location of repressed genes. So any chromosome, of course, has some portions in both of these. It's kind of hard to imagine how these are organized. But here's a representation. Each chromosome will have some DNA sequences in the A or active compartment and some bits of the DNA in the repressed or inactive compartment. And finally, we have the TADs, the topologically associated domains, which form these large loops that organize the DNA in ways that will actually, very importantly, impact regulation. Okay, so we're going to say quite a bit about TADs as far as their roles in organizing chromosomal DNA and in regulating gene expression. Okay, they're very, very important. And we're going to talk a little bit about one of the methods that's used to observe these kinds of long-distance DNA organization arrangements. And this is called Hi-C, which involves cross-linking the DNA and determining what contacts individual sequences have along large regions of the genome. So this is chromosome conformation capture Hi-C. Okay, so the idea is that DNA sequences that are close to each other in the linear genome will interact with each other, of course. But many times the DNA will actually interact with other DNA that's really quite far away from it. And this has important implications as far as regulation. So we're going to spend a little time just describing one form of this incredibly interesting method to look at chromosome organization. Okay, so here's the TAD idea. We have organization of these megabase long loops of DNA with the help of some particularly important proteins, one being CTCF and the other cohesin. CTCF is a DNA binding protein, and cohesin is considered an architectural or structural transcription factor, and they're both very important proteins. Okay, so we're talking about a Hi-C map. Okay, the basic idea of this technique, without the analysis part, the DNA is cross-linked, to whatever molecules are nearby itself. Then, in order to see what was interacting with who, we cross-link the DNA. We take the cross-linked DNA, cut it with a restriction enzyme that creates small pieces, and then dilute it so it can only ligate to something that's connected to itself. So naturally, most of the cross-links will be to DNA sequences that are normally near each other in the chromosome. But some will be to sequences that are not at all adjacent, in fact can be megabases away, but interact sometimes in physical proximity as a result of the chromosome organization. So ultimately, we end up with a gigantic amount of sequence information, and the contacts will be displayed as a map. And the appearance of the map depends on what distance range we're looking at. Okay, so the Hi-C method starts off with cross-linking DNA, and that two pieces of DNA that are in physical proximity to each other will remain there for the duration of this analysis. Then we cut with a restriction enzyme, fill in the ends of the restriction enzyme, marking them with biotin, very importantly. And then we will ligate them, release the molecules, and then shred them, and then sequence them. Sounds not too complicated, but it sort of is. Some of the important points, however, have to do with the labeling with biotin and the organization of the sequence when you actually get it. Okay, so we take our cross-linked DNA, and we're going to cut with a restriction enzyme. In this case, it's going to be ND3. Okay, so we need to label these with biotin in such a way that we're going to be able to recover the labeled ends later. Okay, so I'm doing a fill-in reaction on this cut ND3 site, and the labeled C is going to be attached to the biotin. So having done that, we can then ligate the DNA to itself, and that will actually form circles. Okay, so what the labeled CTP labeled biotin looks like is this. It's hard to imagine that the polymerase will accept something that looks like that, but it does. And so the occurrence of the biotin at that part of the molecule will mark it for later manipulations. Okay, so we're going to ligate the molecules in very, very dilute solution, so that the only ends that can ligate are ones that are already near each other. So this will attach our two pieces of DNA to each other with the biotin label in the middle. So then we're going to reverse the cross-links, shear the DNA, and importantly, purify the biotin labeled pieces. Okay, so we're going to do a pull down with biotin using strepavidin, which has a very strong affinity for biotin, and that allows us to purify the biotin labeled DNA fragments. Okay, so then we can sequence with paired ends, and we'll be able to determine what sequences were adjacent to each other. Now notice in this picture you've got the blue piece of DNA and the orange piece of DNA, and they're now attached together. Now those were not near each other in the genome necessarily, and so having them attached to each other with the biotin in the middle allows us to determine where the blue DNA and the orange DNA were actually in proximity in the original cross-linked molecule. So the junction of the ends is important because it tells us what those two pieces were in contact with each other. So they may have been physically close together, but far apart on the DNA molecule. So it's going to take a lot of sequencing reads to get this all sorted out genome-wide. So for those of you who want to know a little bit more about how we know some of this stuff, what's represented here is a chromatin conformation capture map of one part of one chromosome. And that's to understand something about how we know where these topologically oriented domains are, associated domains. Okay, so in this picture, we have chromosome 12 written along the top in megabases. Okay, the same chromosome is on the vertical axis. So what we're showing is where the DNA at each location in the chromosome actually contacts other DNA by methods that we're not going to really go into. Okay, now it makes sense that the strongest signals would be along the diagonal because each sequence in the genome is most likely to contact something which is very near itself. That certainly makes sense. What makes a little bit less sense is if you look further away, where you can see that there are DNA contacts between and among sequences that are really relatively far away from each other in the megabase range. So that's where we get the idea of topological associating domains and how we can look at them. Okay, so this is represented in the expanded view of a 2.8 megabase region along the chromosome, where the contact frequency is represented. And you can see that within this 2.8 megabase region, a particular DNA sequence can interact not with something which is right near itself, but by something that is a megabase or more away from it. And so that's where these contact maps are coming from basically cross-linking the DNA and looking to see what it's attached to, which could be something very close, but it could be something relatively far away. And that's what these little squares are. I think this is what I just explained. The little red dots mean that the sequence at the top interacted with the sequences along the side. So sequences near each other, of course, interact very frequently. But what's impressive here is that if you look on the diagonal, some things that are relatively far away from each other are still able to be physically near each other in the actual folded state of the chromosome. So here's another picture of our contact map. We already said this. Okay, this picture actually says a little bit about how these loops can form, and we'll get to that in a second. But the important things for this is that the domains of the chromatin are held together in the form of loops and subloops by specific sets of proteins. And the important ones for us to know are the CTCF protein, which defines the borders of these regions, and the acohesin complex, which actually encircles the DNA at the sites of interaction. Here's another representation of these TADs, which is maybe a little bit easier to look at, where we have complexes of CTCF and some other proteins sort of scattered along the chromosome, along the DNA molecule. But those regions can be brought together along with the cohesin complex in order to isolate the DNA between them in the form of a loop. And so we can represent those loops as contacts between those border regions where they are actually brought into the loop form. And there are even some contacts, contacts within the TAD as part of it. So these different forms that these can take are represented by the interactions. The two red things are brought together, but you can have sub-TAD compartments as well. Okay, so the point of it is DNA sequences that are fairly far apart are brought together to form loops, and these are the TADs. Within the larger TADs, there can be many sub-TADs in which the smaller regions within the TAD are also brought together into loops. And that is often for the purpose of enhancer promoter interactions. Okay, and the most well understood of the proteins involved in this are the CTCF protein and cohesin. So here's a representation of a TAD and another TAD where we have the CTCF bound at the TAD boundary, and that anchors the loop, the two ends of the loop, and restricts the interactions of DNA within the loop, within itself versus looking at other loops. There's also sub-TAD compartments where proteins within the loop can be brought together to form a subloop. And these also involve CTCF. Okay, so we can envision our genome as consisting of clusters of neighborhoods, if you will, that are isolated from each other by the combined interactions between these proteins and also many others that are not always represented here. So CTCF is a CCC-TC binding factor, and it is found at TAD borders, but also within TADs, where they serve different purposes, involving transcriptional activation many times. Okay, there are many, many thousands of binding sites for this protein within the genome, and one of those involves loop formation, but also for transcription activation and repression. The other important protein complex here are the cohesin complexes. Right, so these are circular complexes, and they are made of SMC, or structural maintenance of chromosome proteins. Okay, there's not just one type of cohesin, there are other types as well, and they play many roles in the cell. Okay, the important thing is that they can form circles, which will then encircle the DNA, and the best known example of what cohesins do involves keeping the sister chromatids together after DNA replication, but they're also involved in organizing these TAD loops and TAD domains and subdomains. Here's another picture. They form ring-like structures to encircle the DNA on the chromosome. Okay, they're also key to forming TADs and DNA loops, and they're actually able to encircle the DNA, but also thread the DNA through themselves to form loops in what's called the loop extrusion model. So here's the idea. They can encircle the DNA and then move the DNA along, and then in this particular example, bringing the little red dot and the little blue dot together to form a loop. So the cohesins themselves do this in an energy-dependent process in order to actually form these loops. Okay, so TADs, the TAD boundaries, what are they actually doing? This represents an enhancer and two promoters, one on each side. Okay, the blue thing is the enhancer, and the little boxes with the arrows represent the promoters of nearby genes. Okay, in this scenario, we could have this enhancer. It could actually interact with either one of those promoters and activate it. Okay, when we put that enhancer in the middle of a TAD, things change. The blue thing now has got a CTCF binding site to its right between separating one promoter from the other. So that little stop sign represents a CTCF mediated border of that TAD. So as a result of the presence of that TAD border, the gene on the right is not activated by that enhancer. It's essentially insulated from it. Okay. Here's a representation of what can happen or what will happen in a wild type versus mutant situation. Okay, so at the top we have a little stop sign, which refers to CTCF, and it has a gene represented on the left and an enhancer located on the right. So the negative arrow indicates that presence of that binding site or TAD border is going to prevent the enhancer on the right from activating the gene on the left. But in response to mutations, some things can change. For example, if that binding site is removed, then the insulator function is basically lost. And as a result of that mutation or loss, the enhancer can activate the gene to the left. So having a TAD border is a way of insulating enhancers from the genes that they could potentially activate. On the bottom is a similar idea that the TAD was disturbed by the inversion of a DNA sequence, which put the TAD border stop sign to the right of that enhancer. And in that situation, the enhancer then can go ahead and activate the gene that it's not supposed to activate usually. Okay, so there's some mutations that are known like this, but one that's particularly striking is that mutations of this sort give us yet another way to activate proto-oncogenes. Okay, so the first three are fairly familiar with us now, but the one on the bottom represents a cohesin CTCF TAD loop with a proto-oncogene in the middle. Now, if that TAD border is broken by means of deletion of a site, then the enhancer that's immediately next to it can then potentially activate the proto-oncogene that's within the loop. So in the absence of that TAD border with the CTCF cohesin, there is no border and there's nothing to shield that proto-oncogene from the enhancer. So this is yet another way in which proto-oncogenes can be activated. Okay, so there can be severe consequences to these borders similarly. Okay, so here's a famous example of TAD disruption and how that causes limb malformations. Now, this is associated with genetic disease in people, but the same mutations have been reconstructed in mice with similar effects. Okay, so the genes involved, there's 3 adjacent TADs, and the center one is the one that's going to be of concern because it contains an enhancer that is very active in developing limbs. Okay, so the 3 genes involved are the center one, which is naturally involved in limb development, but then its interaction with the flanking 2, which are not normally associated with that process. So here's a representation of the enhancer and the various mutations that have given rise to brachydactyly, F syndrome, and polydactyly. Okay, so the gene in the center is normally associated with limb development. But if you create a deletion where that gene itself is actually missing, and where you take out the TAD boundary that separates it from the next PAX3 gene, what you get is the activation of that gene inappropriately in developing limb, and the result is brachydactyly. Okay, similarly, on the other side, if you basically disturb the TAD border by making an inversion, which allows the adjacent gene to come into contact with the enhancer, you not only get activation of the usual expected gene, but that gene as well. And that leads to F syndrome. The third example is where that region containing the flanking gene is actually just duplicated, and that leads to overexpression of that particular gene, and that leads to polydactyly. So you can have very severe effects simply by moving a TAD border or inverting a TAD border such that an enhancer is brought. Enhancer is then controlling a gene which is not normally associated with that enhancer. Okay, so the bottom line is you have the EPH84 gene, which is included within a TAD, and that includes a limb expression enhancer. So the idea, of course, is that we have adjacent TADs that are not normally under the control of that enhancer, and mutations that bring them under the control will cause these limb defects. Okay, and that includes removal of the TAD border, inversion, etc. So we're not quite sure of the mechanisms here, but the phenomenon is very well supported. Okay, so lastly, we have the idea of Xist spreading and the idea of long-range interactions being involved in this process. So similar ideas are relevant because Xist is going to spread along the entire inactivating X chromosome. And we need to know how it becomes in contact with all those regions of the X. So here's some ideas about how Xist can spread. Model 1, which is very simplistic, says that it starts at its own genetic locus, the Xist locus, and then basically starts off and just spreads linearly across the entire X chromosome. Okay, Model 2 has a lot more to do with reality, where it starts off, of course, at its own, where it's made at its own DNA locus, but it's able to somehow leap to different parts of the chromosome pretty much simultaneously. And that seems to be what was actually happening. Okay, so we don't just diffuse across the chromosome. We go to distant sites early and filling in after that. Okay, so we're going to be basing this on physical proximity to those sites, it seems. Okay, so we're going to capture the DNA that was near the Xist RNA, and that tells us something about how spreading occurs. Now, this is an unusual system. It's an inducible system in which the investigators were able to just turn on Xist RNA in the presence of a drug, doxycycline, and then watch what happens to the Xist RNA as it spreads along the chromosome. So it's experimentally activated in culture cells of early mouse embryos. Okay, and so we can look at its location over time. And the procedure is actually going to involve something called RAP, which is RNA antisense purification. And we're just going to have to take my word for it that this gives us the information that we need. Okay, so we're going to watch over time. We're going to use FISH, and we're also going to monitor the appearance at distant sites along the X chromosome. And we can see if we have no signal. We have not yet induced the Xist RNA. We don't see anything. But over time, 1, 2, 3, and 6 hours later, we get the appearance of the Xist cloud, which is going to be coding the X chromosome. Now, importantly, over time, if you look even early on, we don't have to wait for it to spread to distant sites on the X. They spread almost immediately and then fill in later from that. So we can see from this that the simple diffusion model doesn't work, that early sites are actually spread all over the X chromosome, and then the subsequent more Xist fills in after that. Okay, so we can see what I just said. The fish cloud increases, but importantly, we're not spreading by a simple diffusion mechanism. We seem to be getting to all locations on the X pretty soon, or at least without any real preference to how close we are to the actual Xist locus. Okay, so what this has to do with high C, or chromosome conformation capture, is that we can examine what DNA sequences are in contact with each other genome-wide. Okay, so the gene sequences of interest here are the Xist locus itself as compared to other locations on the X chromosome. So the Xist is close to some locations, but very far from other locations. So we're going to be able to look to see where the Xist DNA is actually located over time. So making a long story short, we're going to be able to see where the Xist is actually located by a method like the high C method that we discussed. And what we can see is that the Xist is spreading to all the sites pretty much simultaneously, where the conformation capture method will matches well the location of the Xist RNA. So this appears to be spreading to many locations at once, partly based on the proximity of the Xist locus itself to those different distant sites on the X. Right, so what this means here, the blue dots are where the Xist DNA is in close proximity to locations all along the chromosome by high C, and that those match the red ones pretty well, implying that the site of Xist transcription, which is the Xist DNA, is brought into physical contact with all those other DNA loci as spreading persists. So one way of envisioning this is you have the Xist locus, which is spitting out Xist RNA, whose job it is to get to all those sites on the inactivating X, and that those sites are being transferred to those locations by essentially a proximity transfer idea, where the different locations on the X are visiting the Xist locus and acquiring the Xist from there. And subsequent to that, of course, the rest of the inactivation process can occur.